Hi everyone. Now we are going to discuss a topic HIV or AIDS from the chapter Human Health and Disease. Let's start discussing this topic. Let's see. From AIDS, let's know what is AIDS. The full form of AIDS is acquired immunodeficiency syndrome. Acquired immunodeficiency syndrome. Okay? Let's see. This is first basically detected in USA in 1981, but in India in 1986 in Chennai. Okay, in Chennai region. Okay, but first time isolated in African patient, and we named it lymphoadenopathy associated virus, that is LAV virus. Okay, after several times we named it HIV. Okay, the full form of which HIV is human immunodeficiency virus and immunodeficiency virus. Okay, it's a family of retrovirus. Okay, member of retrovirus family with a genome size of about 90 to 120 mm. Yeah, 90 to 120 mm. Okay, let's discuss about the structure of HIV, how it basically looks like. Let's see. It's a hypothetical structure. Okay, I'm going to make a hypothetical structure of HIV. Let's see. Like a typical viruses, they have envelope. They have envelope, basically made up of lipoprotein. Lipoprotein is part. Lipoprotein. Okay, and they have the capsid. They have the capsid. Okay, and the most important part they have is they have some receptors. Okay, some receptors that present on their membrane. Okay, these are the receptors. They basically made up of glycoproteins. Okay, they're made up of glycoprotein. For a specific, this bulb-like structure is made up of GP120, glycoprotein 120, and the tract-like structure is basically made up of GP41. Okay, I think that will be clear. Now, what another thing they have? They have two single-stranded RNA. Two single-stranded RNA. Let's write it down here. Two single-stranded RNA molecules. Okay, and they have some enzymes. What kind of enzymes? They also need the enzymes now. They have basically three basic types of enzyme. First one is reverse transcriptase. Two another molecules of reverse transcriptase. First, two molecules of reverse transcriptase. I am writing in short reverse transcriptase. Okay, second one is integrase. Integrase. Third one is protease. Okay, let's make integrase and protease. Let's see, this is integrase and that one is protease. Okay, these two molecules reverse transcriptase, that's integrase, that's protease. Let's have named it right here, reverse transcriptase. Okay, that's it. Okay, now see how retrovirus basically works. That is the most important point here, na? How it will basically work. Let's see. In retroviruses, in retroviruses, they first enters into your body. They first enters into body. Okay. And second, which part? Macrophages part. Okay. Macrophages part. I hope you know macrophages are macrophages. Macrophages and replicates with the help of which enzyme? Reverse transcriptase. This is one of the most important enzyme here. The reverse transcriptase. Okay. And they basically kills messengers. And what kind of messengers? The messengers of our immune system, no? Kills messengers of our immune system. And which messenger? Helper T cells. Okay, helper T cells or helper T lymphocytes. Okay, and how they attacks on T4 lymphocytes. T4 lymphocytes. Okay, when they attacks on T4 lymphocytes, we have certain kinds of symptoms. Okay, we have certain kinds of symptoms like fever, weight loss, and some infections. And we are basically prone to this infections like bacterial, viral, any other type of infections. Okay, we basically become prone to infections like, or infections by what kind of microbes, whether it is bacterial, viral, protozoal, anyone. That's it. Okay, I hope this will be clear. Now let's discuss how the life cycle of HIV or human immunodeficiency virus works and how they replicate. Okay, that's the most interesting part here is, let's see how they works. Let's assume this is our HIV. This is clear, and they have the receptors with it. Okay, that's the HIV here. Okay, and that's our plasma membrane. Okay, and another thing they have, and that's our that's our plasma membrane, that's our nuclear membrane. Okay, I hope this will be clear. Let's see what happens when this HIV okay, this portion of HIV, the GP120 portion of HIV attached to the receptors, let's see, attached to the receptors. What happens? They basically make a way. Makes a way for what? They opens a way for the virus. To release its genetic material. What kind of genetic materials? Single-stranded RNAs. And another thing, single-stranded RNAs, enzymes, certain enzymes, reverse transcriptase, integrase, protease. Okay, that's the first step here: attachment, entry, and penetration. Okay, they first attached, penetrate, entry and penetrate, entry and then they penetrate. Is it clear? Now this reverse transcriptase, the molecules or reverse transcriptase, that's RT, helps them to convert their RNA into DNA molecules, this type of DNA molecules. Okay, is it clear? Now what happens, we have their own genome, we humans have their own genome now. Let's see, we have the genome here. Okay, we have, that's genome here, that's human genome, that's human genome, that's human genome. What happened, the enzyme integrase help this to attach with our human genome. This enzyme integrase help them to attach with the integrase part. Okay, so let's see, now our human genome is basically attached with viral DNA. Is it clear? Reverse transcript helps you to convert RNA into DNA, then they integrate into the human genome, and then the process of transcription occurs. And what happens in the transcription? Basically, when the infected cell divides, the viral DNA is basically read and long chains of proteins are made. Is it clear? Is it clear? Okay. So when this transcription occurs, okay, infected cell divides, and viral DNA read. Is it clear? And the new copies are made, and the budding process is the last and the most important part here. So they come out from the cell with a protein coat. That's clear. This is the life cycle of HIV or replication. I hope this will be clear. So thank you for this. Please like and subscribe.